Welcome to Click for IB Biology's genetics videos. This one's looking at the type of cell division called meiosis and how this brings about what is called the segregation of alleles. And this process leads to the variation that we get in the offspring from sexual reproduction. There's a blank version of this presentation as a PDF down in the description. And also down there, you'll find 10 questions with answers that you can use to consolidate your understanding of this part of the course. So this overview of meiosis will be very useful for those of you studying standard level biology and an introduction to those of you doing higher level biology who should then go on to look at topic D 3.2, inheritance, and the discussion of segregation and independent assortment of alleles. So to the overview of meiosis. So let's remind ourselves that we are dealing with adult multicellular organisms, and their somatic cells, their body cells, are diploid, possessing a double set of human chromosomes, a double set of human genes. And what happens in this meiosis type of cell division that occurs in the reproductive organs is that this double set of chromosomes is reduced to a single set of chromosomes and then thereafter known as haploid cells. So the gamete has a single set of chromosomes, a single set of the genes. Now in this image here, I guess we're implying that this is a human example, but theoretically then, as we've just said, the diploid cells will produce haploid gametic cells. And the interesting thing about this is that the haploid cells, the gametic cells which are produced, are not all the same. Now because of the rules, the ways in which the pairs of the homologous pairs separate during meiosis I, this means that what we're able to theoretically calculate is that the number of gametes that you can get based on the different combination of chromosomes in the haploid is 2 to the power n, where n is the haploid number. So in human beings, the process of meiosis takes place in the male, biological male, in the testes, where the cells are diploid, and they go through meiosis, and we end up with haploid sperm cells. And in the biological female, human female, in the ovary there are diploid cells, and these also go through the process of meiosis to produce haploid egg cells, or oocytes as they're sometimes known. Now, as we said, there's the possibility of creating tremendous variety of sperm cells based on which particular 23 chromosomes come from the 46 which are in the diploid condition. Now, n equals 23, and therefore for human beings, 2 to the power 23, 2 to the 23 is the equivalent for approximately 8.6 million different arrangements of chromosomes in the half set of chromosomes. Of course, that's equally true of the egg cell. 2 to the power 23 means that the biological female is capable of producing 8.6 million different arrangements of chromosomes in her egg cell. And therefore, if any of these sperm cells can fertilize any of these egg cells in a kind of random fertilization process, then we can calculate the number of different zygotes at 2 to the power 2n. And if we apply that to humans, that's 2 to the 46, which I understand to be around 70 trillion different possible fertilized egg cells, or as we properly call them, zygotes. It's an incredible variety just from meiosis creates this incredible variation in offspring without even needing to discuss mutation and without the phenomena which is called crossover. So already we have tremendous variation in our offspring, which of course are subject to the pressures known as natural selection. Let's take a look at the type of cell division which creates this variation. And we have to look at it in a fairly simple example so that we can comprehend what's going on. So here we are going to start with our diploid cell. And this diploid cell is currently in G1 of the interphase. And I'm only illustrating one homologous pair. And so that we can track what goes on, I'm going to identify a dominant allele on this one here. I'm going to call that big A, and I'm going to put little a on this homologous partner here. And the purpose of this is so that we can track what happens to the individual chromosomes as we go through the double stages of meiosis. And that's a point which requires re-emphasizing that meiosis is effectively 2 rounds of cell division. The first round results in 2 cells, and the second round results in 4 cells. The first process is DNA replication, and as a consequence of that, we end up with pairs of sister chromatids, and the number of alleles has already doubled. In the first division of meiosis, known as Meiosis I, also known as reduction division, we have the separation of the homologous pairs. And so you can see that this one here goes this way and this one goes this way. Now, if we track the details of Meiosis I, we would find an example of prophase 1, metaphase 1, anaphase 1, and telophase 1. The ultimate outcome of that will be the separation of the homologous pairs. The second round of meiosis separates the chromatids. Now, if we just hesitate at that point, notice that after the first round, we have separated not only the homologous pairs, but we have separated the different alleles for a gene, and these will end up in different gametes. In meiosis II, also characterized by prophase 2, metaphase 2, anaphase 2, and telophase 2, then to be followed by cytokinesis, we end up with the separation of the homologous pairs, which means that big A would be here and here, little a would be there and there. And notice that this means that 50% of the gametes would be of this type and 50% of the gametes would be of that type. What this diagram does not reveal is the consequences were we to include all homologous pairs. And this is the reason why, and that particular detail reveals the reason why meiosis generates such large number of different gametes. So this is just the beginning of that, but sufficient to be able to carry out standard level genetics crosses. We will explore the more complex version in the higher level videos in D3.2 of inheritance. There's the link to the next video, and of course the subscription to the channel with all the playlists in the genetics topic that we're doing. Thank you for joining us.